It, right? Okay, so I'm gonna have to figure out where I want to stand, where you guys can actually still hear me. Some quick business. So everyone should have received notification about adding this class. If you're here and you haven't added it yet, come see me after. Hopefully all the people who didn't get in are not here anymore, unless they just like to see me. You have until 5 p.m. today to complete the pretest. So do it. Don't miss out on this great opportunity for 10 points. Quiz 1 starts today. Don't forget that. And recitation begins next week. The papers, I'm trying to reorganize my recitation from last year a little bit, so I'm a little slow on getting everything up. The papers will go up at least the Friday before the following week, so you'll have all weekend. And hopefully it'll be even sooner. Okay, so last time, it's so weird, right? I got so used to standing back there, and now I have to stand in front, but then I have to go back here to move it. Okay, so last time we talked about the concept of the cell as a larger part of the organism. This is key, so I'm really trying to drive home the big picture, okay, before we get into the details. So the cell is part of a larger organism, and it's responding to its environment, right? And it responds to the environment by way of signaling. So we're going to go over that briefly in a minute. And that results in gene expression, and that is what this whole course is about, gene expression, pretty much. That response will result in either activation or inhibition, repression of gene expression. And then if you have gene expression, then you're making proteins, and those proteins, their whole job is to function in dealing with whatever message that the cell has gotten in terms of the current environment. Okay? So that's what we've been talking about. And this is where we ended. I put together this kind of workflow, right? You get a signal from the other cell, and it'll either be positive or negative regulation, right? So if there's positive regulation, you'll get reorganization of the chromatin, and then movement of RNA by transcription factors, transcription, processing, translation. Then you'll get some kind of post-translational modifications, which will further affect the protein's function. And then usually when things need to be turned off, then the protein will be degraded. And when you have repression, usually it's kind of tightening up the chromatin or making things go away. So you can't get methylation, perhaps. So you don't get gene expression. Okay, so first, I always realize that I leave this out of this course, and this is really crucial to understanding gene expression, because you need to know where this signal is coming from. So those of you who've taken 408 have a good idea of signal transduction. But those of you who have not taken 408 might need a little refresher. Okay, so the basic point, where's the, okay, there's there like a thing? Hey, I'm gonna be lost in this room for a little while. So the basic point is that you've got some sort of extracellular signaling molecule that's been either secreted by another cell or secreted by its own self, or just something coming through the bloodstream, it could be a hormone, it could be whatever, coming from elsewhere. The whole purpose is it is transmitting a message. And that receptor, there's a receptor on the cell surface, the signaling molecule binds to the receptor, then you have signal transduction, which is just essentially relaying the message to the nucleus to affect gene expression. It's the same thing. I was trying to think of what's a good example? It's like playing telephone, right? Except for the message ideally doesn't change too much. If the message changes, well then that could be your example of cancer, where things go awry and it's a true game of telephone and then you have cancer. So basically, we'll just go through a few examples of signal transduction. I really wish I had a pointer. Come on, guys. Anything? Huh, what's that? Could there be a pointer on here? What do you think's going to happen? This is all Elmo. Oh well. Okay, I'm just going to complain later. Okay, so here's your generic signaling pathway, and those of you who've had 4-8 with me will recognize this one's the next, I think, 3 slides. This is a very generic signaling pathway. At the top, you have a signaling molecule that's bound to a receptor, and then there's this relay of proteins from the cell surface all the way to the nucleus, and their whole point is to pass the message along, usually in the form of phosphorylating each other, creating complexes and phosphorylating something else, and all the way down until you have some kind of messenger protein, which we will refer to as the transcription factor, which will enter the nucleus. And the whole point is making that transcription factor able to enter the nucleus. So normally it's excluded from the nucleus because there's no nuclear localization signal, but when you get either phosphorylation or something that causes a change in conformation, suddenly there's exposure of a nuclear localization signal and the protein can go into the nucleus. So the end result is it goes into the nucleus, it binds to some promoter, recruits RNA polymerase, and you have gene expression of the specific gene that needs to be turned on. And this is, when you talk about cancer, it's all about signaling. Well, not all, but there's a lot about signaling. And usually when certain players in signal transduction pathways are mutated or overexpressed or deleted, depending on what their job is, that often is involved in formation of cancer because you get dysregulated signal transduction. Okay, so I'm giving you guys a few examples, right? So these are different processes that affect gene expression in response to some kind of extracellular signal, usually coming from another cell. These are examples. Do not freak out. People freak out when I show signal transduction pathways. Do not freak out. I do not want you to memorize them. I don't want you to know them. I want you to understand the concept. Okay, so I'm showing you, I think, 2 or 3. This one is the JAK-STAT pathway. It doesn't matter. It's just an example of one pathway that is involved in the immune response to viral infection. So you have some receptors. Oh, I wish I had a pointer. So you have receptors. They're on the cell surface. And you have your signaling molecule, which in this case is alpha interferon, the red circle at the top. And that's secreted by some other cell in response to viral infection. And the whole point is to alert the cells in the area that there are virally infected cells. Is that important? Do you want to alert the area that there's an infection? Of course, right? That's how you mount an immune response, right? So without this signaling, without these molecules being secreted from cells, these proteins being secreted and binding to receptors, we're not going to have an immune response and we would be gone as a species. So you have the signaling molecule, in this case alpha interferon. It binds to a receptor, which causes a bunch of changes in conformations, right? You have dimerization in this case. You get phosphorylations. You get recruitment of other proteins. Notice I'm being purposefully vague because I don't want you guys to get bogged down with the details. But the whole point is you have these changes that allow, in the end, proteins to enter the nucleus. In this case, the STAT dimer has a nuclear localization signal that is now exposed. You can see this down at the bottom here. And it can go into the nucleus and it binds to some promoter and activates gene expression, and that's involved in expression of different genes involving like cytokines and stuff like that involving the immune response. So here's another example, right? Notice it looks exactly the same, right? You've got the cell surface and some kind of receptor, some kind of signaling molecule in red, TNF-alpha up top, and it's going to bind to the receptor. There's going to be a bunch of things that happen because the binding of the signaling molecule to the receptor causes trimerization of the receptors, so they all cluster together, and that creates different binding sites that allows different cellular proteins to be recruited to the cytoplasmic domain of the receptor. And then there's all this phosphorylation, and all this stuff happens that in the end results in the ability of NF-kappa B. So normally NF-kappa B is inhibited because it's got an inhibitor bound to it, and signaling results in it going away, it's degraded, and that liberates NF-kappa B to go into the nucleus, bind to a promoter, activate gene expression. Okay? So I think that might be, okay. So you might be saying, what's the big picture here? That's a whole lot of signal transduction pathways that are freaking out. Hopefully no one's freaking out. People tend to freak out early in the class because they feel like it's a lot of, is anyone freaking out yet? No? Good. Okay. If you feel like you're freaking out, you should stand up and go, ah, so I know. And that way I'll like tone it down. No, I probably won't do it. Okay. I was like, come to my office hours and we'll talk through it. Okay. So, what is the big picture here? What is she talking about? Right, so I'm talking about gene expression, and I really want you to understand it's occurring based on something. It's not random. It's due to an outside signal. Where's that outside signal coming from? Right? So the cells, they need to respond to the environment, and these signaling molecules that are secreted from other cells are the way of transmitting a message to the other cells in the area, and this results in signaling transcription factors that enter the nucleus, and what? What are these transcription factors doing? Inducing gene expression, right? Because they're binding to a promoter, recruiting RNA polymerase, then you get gene expression of whatever genes are important there. So you should understand that there are, I don't know, how many signal transduction pathways do you think there are? Do you think there's like 5 or 500? I'd say in the larger number, not 5. There's so many, and then they merge at some places, and they separate into different areas in different places. There are canonical and non-canonical formats of different pathways. I just want you to understand it's not just as simple as this, and that complexity is what makes us such complex beings, right? There's so many different pathways, so many molecules that transmit so many signals that activate many different genes under a variety of situations. Okay? Okay, so I keep talking about transcription factors, transcription. What is transcription? Who wants to tell me? Oh God, you guys all have to leave if you don't answer. Yes?
DNA codes converted into RNA.
Right, so DNA to RNA, that's transcription. Right, absolutely. Okay, so when does transcription happen? In response to environment, when RNA polymerase binds. Okay, so when there's some kind of signal, does it happen after DNA replication? Okay. I once had one of my smartest students ask me if you always had transcription after replication. And I wanted to kill him because why would you copy your whole genome before you express one gene? It seems so inefficient. But it's taught that way. You know, in 201 or 309, people do replication, transcription, translation. So people think you need to replicate your DNA before transcription. If you think that, you don't have to tell me. But if you think that, just remove it from your brain right now. It happens, transcription only happens when a transcription factor goes into the nucleus and activates transcription of certain genes. That's the only time it happens. Okay, so how is it regulated? How is transcription regulated? What do I mean? I'm going to use the word regulation a lot. So regulated, I mean, what turns it on, what turns it off? Repressors and activators, right. So usually there'll be some kind of activator, maybe there'll be a transcription factor, or maybe there'll be a repressor that will come and bind somewhere that will prevent RNA polymerase from being recruited. Okay, so what's the outcome of transcription? Expression. So what's the next step? So you've got transcription, then what happens? You guys should raise your hands. Stand up and be proud of your answers. Yes?
RNA processing.
RNA processing, right. Okay, so give me an example of an event that would result in transcription. You guys want to talk to your neighbors for a minute? Take a minute, talk to your neighbor, and come up with some example of something that would result in transcription. Okay, guys. I like to give you a few minutes to chat, but then stop you before we start talking about the Super Bowl. Okay, who's gonna watch the Super Bowl? Of course. Do you think that you're gonna be kicked out of Maryland if you don't watch it?
What?
Do you feel like you'll be kicked out of Maryland if you don't watch the Super Bowl?
I don't know, this guy was yelling at me just like a couple minutes ago.
Said he wanted the 49ers to win.
I don't.
I have to tell everyone.
I totally don't. Is there gonna be a fight? I went to a Yankees game. I went to a Yankees game with a guy who was wearing a Red Sox hat. That was bad, right? So we don't want any of those fights in this class. Okay, so someone give me an example. Yep. Immunizations, absolutely, right? So immunization, anything that's gonna elicit an immune response is gonna have to result in some kind of gene expression because all those immune cells are gonna start secreting cytokines. Right? Very good. What else?
Pregnancy?
Oh yeah, pregnancy. Right? Okay, so specifically what are you thinking of? Somebody
Well, the hormones that tell the uterine wall not to strip or, to push out that foreign object.
Okay, all the hormones involved with pregnancy that make you not get rid of the embryo, right? That's actually, oh God, whoever has a chance to take immunology should take immunology because that's an interesting one. If you think about it, right, we reject foreign organs, but we don't reject our babies until delivery. Then we reject them. That's essentially a form of rejection, but that's another story. No, I'm not being funny. I'm serious. Okay, and I always like to give the example of placentas, right? There's got to be some gene expression that results in the growth of a placenta, right? Because would you grow a placenta if you didn't need one? Like, what if we just randomly started growing placentas in our uteruses? And men, what if men started growing placentas? I don't know, where would you grow them? Okay, good. So we all understand the big picture? Okay, so here's my example, right? So you're evaluating the response of B cells to the presence of bacteria, right? So what kind of genes do you think would be transcribed in the presence of bacteria? Someone raise your hand. Yes. Antimicrobial peptides. Why not? What else? That's it? That's it? So you have a bacterial infection and you make a few antimicrobial peptides. That's it? We would totally know to run this species. Yes? Okay, so I'm hearing inflammatory response. So cytokines, right? So you need to start transcription and translation of cytokines that are going to be secreted and then activate transcription in other, activate gene expression in other cells. Yes? Apoptosis genes, maybe for viruses for sure. But maybe there might be some effect on apoptosis genes as a logical assumption. Yes? Antibodies. That's number one, right? First thing you want to do, if so, that's what's showing here. You'll have your macrophage, which has engulfed your bacteria, and then it is showing off a little bit of actually. Interesting. Anyways, so your B cell antibodies are going to recognize the bacteria, and you're going to do a whole bunch of things, one of which is start growing, right, and start dividing and change your antibodies from membrane-bound to secreted and start having tons and tons of B cells secreting the antibody that's specific to the bacteria. That's part of the immunization response there. What else? Yes?
Increase production of white blood cells.
Right, so cell cycle. Cell cycle is going to be turned on because you want to produce more white blood cells so you can fight the infection. So that means you need transcription and translation of things like cyclins that are active and activate your CDK so you can go into cell cycle. Okay, good. Anyone else want to add something? I think those were some good ones. Okay, so how do we study gene expression? So I'm going to give you guys 4. I think we're only going to cover 3 of those today, but there's 4 methods that we'll go over that are common for studying gene expression. Northern blot, microarray analysis, reporter assays, which I believe I'm covering on Monday, and real-time PCR, which is quantitative PCR, so you can actually get a number of transcripts. Okay, so we'll start with northern blot. Who's heard of a northern blot? Oh, good. So you guys are going to be bored for the next few minutes. Okay, so a northern blot you can use when you know what you're looking for, because your probe that you design is specific to your gene of interest. Basically, what a northern blot is you're just trying to detect some kind of RNA. So you think beta-actin RNAs increase. So that's usually loading control. But anyways, you think beta-actin RNAs increase under certain condition. You design a probe that is complementary to the RNA sequence for beta-actin, and you'll detect it on a membrane. So basically, you isolate all your RNA from your cell where you think that RNA is, and you do electrophoresis to separate your RNAs based on size, and then you're going to transfer it to filter paper or membrane, actually, and then you incubate it with your probe. So this is your probe that's specific to beta-actin, and you wash it, and then you expose it to X-ray film because it's usually radioactive, and you get one band that would be your beta-actin band. And the reason it's only your beta-actin band is because your probe is complementary to the beta-actin RNA sequence. Okay? I have a little link at the bottom for a video that you guys can watch if you want to understand it a little bit more. So this is useful in some cases, right? It's useful when you know what you're looking for. Sometimes you don't know them. You know that sometimes you want to discover genes that are being induced. You can't do that here because your probe, you have to design your probe to the gene that you're interested in. Traditional use is radioactivity. Some people don't like to work with radioactivity, especially if you're pregnant. Just so you know, it's a bad thing to do. You can use fluorescence also. So there are better ways to do these things. You can identify differences in size of transcript and variation. So let's say instead of one band in the end, you get many bands. What might that be? So now you're seeing many different bands of different sizes, but they're all hybridizing to your beta-actin probe. When you have RNAs of different size, what could that be due to? Folding? No, well, actually, so when you run your RNA on the gel, it's kind of linearizes? A good question. Yes?
It's cross-reacting with a bunch of different RNAs. So
Well,
maybe
it's definitely-
not specific.
So it is definitely reacting with a bunch of different RNAs. The question is it specific or not? So one thing you should always think of, did I design a bad probe? And is it cross-reacting, and am I getting a bunch of garbage? So let's say you take that probe and you blast it. Does everyone know what blasting is? Blasting is when you compare the sequence of something to the whole genome. You can do it, there's the databases online, and you put your sequence in, you hit blast, and it will pull up every instance of that sequence in the human genome, or whatever genome you choose. So you blast it, and you see it only pulls up beta-actin. So you're like, okay, well my probe is good. Why else could you be getting a number of transcripts of different size? Yes?
Could it be that some of it's denatured?
So that's like what I heard over here. Some of it's denatured. Well, so when you run a gel, usually you don't retain secondary structures. So what I'm looking for is there are three. Did you have one?
The gene is polymorphic.
So the gene is polymorphic. So yes. So that's the direction I'm going in. There are polymorphisms in genes. But in an individual, usually you'll just have one version, but what if you have different splice sites, right? And you have splice variants, right? So if you have splice variants, that means there are different splice sites, and sometimes you'll have one splice site that's used, and sometimes you will result in RNAs of different sizes, right? So that's where I was going with that. Okay. So what kinds of questions, so it's great that you know what a northern blot is, right? But how do you use it? What's the point? What kind of questions can we answer? So pretty much anything that requires detection of a specific RNA. If you're looking for a certain RNA, you can do a northern blot. Now mind you, there are way easier ways to do it than a northern. So just for all of you guys going into research, northern should be down at the bottom of your list because real-time PCR is way easier. But this is still important to understand. So you're studying the role of a viral protein. You're studying the role of a viral protein in exporting unprocessed RNA from the nucleus. Now this requires you to understand that some viruses, they make transcripts that have multiple coding regions, and they'll export the whole thing from the nucleus. And depending on where the ribosome binds, you can get multiple proteins. HIV does that, for example. And so this virus needs to export an RNA which contains introns, which is not processed out of the nucleus. And that shouldn't happen. So a lot of viruses, they'll encode their own proteins that will bind to RNAs and help them get out of the nucleus. Okay? So you know that virus X synthesizes multiple proteins from an unprocessed transcript, you know this, and you know the size and the sequence, but you don't know which viral protein is responsible for exporting. So what kind of experiment you would do, and what results do you expect to see? So I know this is a lot for the first week of class, but I'm just going to challenge you guys to think about this for a minute. So talk to your friends. Do you need me to expand on the question at all? Or are you good with the question? It's right up. Does anyone need me to clarify? I know it's hard to think experimentally sometimes, but we're going to get started quickly on this. Okay. So the figure that I have here is showing the northern blot with, it's a result, right? Where you have cytoplasmic and nuclear. So you can actually take a cell and isolate the nucleus and the cytoplasmic fraction, and you can detect which RNAs are present in which place. And that's useful for answering a question like this because only certain viral proteins are going to allow your RNA to get out of the nucleus, right? So if you start to think about viruses and how you can delete proteins from viruses, you can do a lot with viruses, especially small viruses. And you can start to use these tools. Use a northern blot because you know your transcript, you know its size, so you can design your probe, you know how to separate your nuclear and cytoplasmic fractions, and you can delete certain genes. And somehow you should be able to determine which viral protein is responsible for this transcript getting out of the nucleus. So talk to your friends for a minute. It's okay if you're like, I have no idea what you're talking about. But I want you guys to get used to thinking like this because these are the kinds of questions that do show up on my exam. Okay? So, check. Oh, you do not know which viral protein is responsible for export. So basically, what can you do to find out? And what would you expect to see?
Because if you knock out that gene, still be
No. Well, no. So that's so that's your real life. You can do whatever you want. Yourself,
So if you isolate the outward, you can see that.
Okay, there's a lot of quiet. There's a lot of quiet in here. I definitely hear a lot of inspiring conversation, which is good. But I see a lot of quiet, confused looks. So we're going to work through this together. So does anyone want to tell me what they think we could do in order to determine which viral protein might be responsible for export of an unprocessed transcript from the nucleus? Yep.
Well, since you can specify your probe to a certain RNA, I was thinking maybe you could somehow isolate the RNA as it's being exported so the protein's attached to it, and that complex would still interact with the probe, and that would give you the size of the probe.
Yeah, that would not be a northern blot, but that would be a way to do it. Okay, so you're saying isolate your since you know your RNA, find out a way to isolate your RNA and see what protein's attached to it. Yeah, I think that would be like a chromatin IP almost, kind of, but not really chromatin, but yeah, I can see if that would be a logical way to approach it. What about a way where you're using a northern blot? Yes?
We came up with, after you see which proteins are in the cytoplasm, that since we already know the transcript of it, you can like find some way to knock it out so that it's not expressed, and then see if it will still occur in like the cytoplasm, you know, if it had role in exporting it out of the nucleus.
Okay, so knock out viral genes that might be involved in export and see if it's still exported, right? Okay, so does anyone want to add to that? So that's exactly what I was going for. So here you're seeing what was in the cytoplasm, what is in the nucleus. So you see this is, you know this transcript is what's exporting, you just don't know how. So if you make your probe and you do your northern, you see this in your cytoplasmic fraction and all this stuff in your nucleus, which is probably just different splice variants or whatever, because viral proteins tend to do that. Viral genes tend to do that. But then what you can do is you can knock out individual genes in your virus, especially the HIV virus is super tiny. People do that all the time. They just one by one knock out the genes and see what changes. So if you knock out gene X, and what would you expect to see if it was responsible? So I heard a few things. What did I hear from over here? It wouldn't leave the nucleus. So would you see anything in the cytoplasmic lane? Right, you would see that band at the bottom would disappear. Anyone else want to add to that? Anyone have questions about this little exercise? I think sometimes when you just learn about a method and don't see how it can be used, it's not very useful. Then you're just like adding it to this laundry list of stuff. Okay. Okay, so now we've got real-time PCR. So northern blots are super old school. Why would you want to do all this, run the gel and make this radioactive probe and all that, when you could just do real-time PCR? And the way you do this, does everyone know what PCR is? Okay, does anyone need me to do a quick refresher on PCR? I can do it, yes, thank you for being honest. So basically all it is you are, you're melting it's a photocopy machine for DNA, right? You've got your double-stranded DNA, and you heat it up, and you melt it apart, and you've got primers that are specific to whatever gene that you want to amplify, and they bind to, you can see right here, so you heat it, they separate, you've got primers that bind to whatever region, and then you allow them to anneal, and then you have DNA polymerase in your tube, and as you change the temperature, you have the DNA polymerase actually polymerizes DNA, so you've got replication of that strand of DNA. And so each cycle of PCR, you get another copy, another 2 copies, right? So you start with 1 DNA, you melt it, and then at the end of that cycle, you've got 2 copies. And then you go from 2 to 4, and it's actually an exponential increase. But with real-time PCR, so PCR is great if you just want a really dark band. But if you want to be able to quantify something, you need a way to do that. And so with real-time PCR, you're actually using some fluorescence in a computer to detect the fluorescence. And you're able to quantify how much transcript you have based on the fluorescence intensity. And I'll go into this. So basically, you need to have a control and some experimental conditions. So it could be cancer, tumor cell, normal cell, right? Because tumors express genes differently. And you're interested in some growth factor gene, right? Because tumors grow quickly, so they probably have some higher expression of growth factor. So you'll treat your cells, or whatever you'll do to your cells. Let's say it's just normal tumor, don't treat them at all, and you isolate all the RNA in each cell. And you make cDNA, so you're copying it into cDNA, but using a reverse transcriptase. And then you do your PCR, and you can quantify how much transcript there is. So first you isolate all the RNA from your cells, and the purpose of this is just to make it into DNA. RNA you just don't ever want to work with RNA. It's not very stable, and also DNA polymerase doesn't use RNA as a template. And you use reverse transcriptase to make the cDNA copy of the RNA. So let's say you have RNA that's very expressed at very high levels. There was really high transcription of this growth factor in your tumor cells, and there's 5 million copies of that mRNA. Well, when you copy into cDNA, you'll get 5 million copies of that cDNA. Let's say in your normal cell there's only 1 copy, you'll just get 1 copy. So it completely relates. And then you'll do your PCR with your specific primers, right? Because you only want to amplify the gene of interest. It's similar to the northern, where you have your probe that's specific to your gene of interest. Here you've got your primers. And it's the same thing. This is just showing one cycle where you have your double-stranded DNA, they denature, you've got primers annealing, you've got your polymerase, which is going to bind, and then you've got copying of the strands by your polymerase, and then you're going to then denature again and again and again and again, and that's how you get your expansion of your DNA. So this is showing you one cycle. You're going to repeat this for 40 cycles. And so with real-time PCR, you'll use some kind of fluorescence, either cybergreen or TACMAN, it doesn't matter, but in this example, it's cybergreen, and it only binds to double-stranded DNA. So you can actually follow how many copies you have by the intensity. If you've got 1 copy of DNA, it will be a lower intensity than if you have 500 copies of DNA. Does that make sense? Right. So you've got a computer that can detect this. It's all being done in the machine. So there's the iCycler. If anyone wants to buy one for me, I'll give you an A if you buy one for me. It can be bought. They start at like 18,000, but I think you guys student loans chip in together. No? Okay. Is that wrong? Would that be wrong for me to have you guys buy me lab equipment for A's? Okay, I think that is wrong. Let me go back in time. I never said that. Okay, so you get this exponential increase. You get this exponential increase in cDNA as your cycles increase. So this is just an example. Cycle 0, you've got 1 copy, and then by cycle 34, it's a lot more. Right? Here's your increase. Right? So that gets plotted on a graph by the machine. You're not plotting anything, and you get the readout is something like this. So this is just an example of different dilutions, just to show you your starting number directly correlates with your ending number after 40 cycles of PCR. Kind of like who's taken micro. So you guys understand the concept of a bacterial growth curve and all that stuff. So let's say you inoculate your media with 1 bacteria. It's going to take a lot longer to get a high optical density of bacteria compared to if you put in 5 mils of a really concentrated stuff, right? You can see how your starting concentration of bacteria or DNA in this situation relates to the end result. So you'll go through 40 cycles of PCR, and then you can tell by the fluorescence. The fluorescence is gonna follow like this. It's gonna increase with your amount of DNA, and then you place an arbitrary threshold, which will be this line, which actually should be up here, and you'll say, oh, well, this one crossed the threshold at cycle number 17, and this one crossed at 36. So this one had a lot more starting material than this one, because this one, the fluorescence increased really rapidly, whereas this one took a really long time to get to the same level. So we will start, I know we're done, but we'll start right here Monday. Don't forget the quiz.